Speaking of which, from last week, the exit survey was, how do you feel that the pace of class has been so far? This was anonymous, so maybe you were saying what you thought I wanted to hear, but that's a pretty good balance. Apologies to the people that think I'm going too slowly or too quickly, but if this is accurate of everybody's feeling, then that's pretty good, I think. Most people say it's about, although what you mean by about right may not be what somebody else feels is just about right. So I'm going to give you one other chance, at least now, one more chance, to practice a gapped alignment, which we talked about last time, which was sticking hyphens in the middles of sequences to better align multiple motifs that might be inside an alignment. The other reason I wanted to do this is I think this version might record. Last time when I did an example, it was on my laptop, which wasn't recorded in the movie, so I wanted to do one where the answer will show up and be recorded in the movie. So you haven't seen this one before, unless you've looked at the slides in advance. Do we see any motifs that look like we could line them up?
AAC.
So I hear AAC. So there's a GAT there, so we could line up the GAT and see if that's a motif. This is always an exploration-based thing. You sometimes don't know which one to focus on first. So let's see. If I'm going to line up that GAT, what should I do? Slide this top line how many letters? Two to the right. So if I insert 2 hyphens, hey, it's working. Technology for the win. Okay, so 1 and 2 line up pretty well. Okay, then what about 3?
3 hyphens at the beginning.
So we can line up the TAA maybe. Except this font is not ideal for lining everything up. But so TAA, CAT, CAA, pretty close. Okay, and then the 4th line, I wonder if spaces are better. Nope. We can line up the AAC. So we've got a motif there, AAC. What next?
There is a CAT motif in all of them. And
Cat.
No, not in the third one. Well, the third one has CAAT, so there must have been an insertion in that one.
Could be. What about gap? Is that a motif? You could try lining them up on that. Let's see. Let's try lining up the GAT. By inserting some hyphens in the 3rd sequence. How's that looking? Let me try to space these out a little bit better. Ha! How's that? Is there anything else that could be done to make that better? I know it's hard to see with the font widths not being identical between the different characters. So we've got GAT, 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 GAT. It looks pretty good. Maybe it could be refined more, but that's the point of a multiple sequence alignment. This is definitely the type of answer where there is not one right answer. There could be multiple right answers. There might be one best answer, but there's some good answers. So in this case, are those 2 hyphens in the 3rd sequence an insertion or deletion? So we've got CA on the left of this TA. We've got a TA in sequences 1, 2, and 4. So there are more ATs in that spot than there are deletions. So it might be a deletion instead of an insertion. Remember, when you're answering questions, this is your interpretation of data. There may not be a right answer, so what I'm looking for particularly is, can you justify why you made the decision you made? When you do multiple sequence alignments, sometimes there are multiple different ways to align the sequences, which is fine. I'm asking you to explain, if you feel like you're not sure if you got the right answer, explain what you did to get the answer you arrived at. Any concerns or questions about gapped alignments? We're adding spaces or hyphens or some sort of gap character to better line up different motifs that are spread out throughout sequences. I told you I was going to report back. Somebody last class asked a really great question about breast cancer. And remember, we were talking about mutations. It's a topic for the class right now. And how mutations are solved by, or fixed, corrected by enzymes that we have in our cells, like the BRCA1 protein. It's a DNA repair enzyme. Its job is to go fix DNA, usually double-stranded breaks, when chromosomes have broken. And so a great question was asked, why is the breast cancer 1 gene susceptibility particularly to breast cancer, not other forms of cancer? But I did do a little literature search and found this fantastic review paper, right, not primary literature. It doesn't have any primary data in it. This is a paper that's all references to other studies that have the data they summarize. But they note that these mutations are indeed found in other types of cancers, not just breast cancer, sometimes prostate cancer, and other forms of cancer. So BRCA1 mutations aren't just predisposing to breast and ovarian cancer. They can be found in other forms of cancer. And so this review concluded that it's unclear. So we still don't know really why it is that mutations, the disabling of the BRCA1 protein, leads often to breast cancer and not much other forms of cancer. But I did find this paper, which has an astounding number of authors on it, which is the way science works these days. You get multidisciplinary collaborations between people in all sorts of institutes and centers and universities. So this is a really recent one, about a little over a year old, a year and a half old. They found that it's more often the case that it's a particular type of cell that's susceptible to breast cancer, cellular proliferation, the formation of a tumor, and those happen to be down at the bottom there, cells that have something to do with genes that are expressed related to estrogen response. In other words, it seems to be tissues that are regulated by hormones, like estrogen, are the ones that are more susceptible to being tumor forming when they don't have BRCA1 to repair DNA damage. So that may be the real root of we still don't know the exact answer for why breast tissue more than other sorts of tissue, but breast tissue has, my understanding, is that breast cancer has more luminal epithelial cells than other types of tissues. And if you have more of those types of tissues, you're more likely to have tumors form in those tissues if you have that BRCA1 mutation. So there may be I think maybe you suspect that there's a relationship between which sex you are and whether or not you're producing estrogen. And if this is an estrogen responsive cancer, then it's more likely that you would get it if you're female than male.
Did the papers mention anything about people on estrogen, like pills, or people taking hormone?
I didn't see that in this paper, but that's an excellent question. So yeah, would you be more likely to get breast cancer if you had this mutation and you were taking exogenous supplemental estrogen, for example? I don't know.
Combined contraceptive, like pills, whatever hormone birth control, there is an increased risk sometimes to get breast cancer, I think, but not ovarian cancer, something like that.
That's interesting to know. So is there a relationship between taking estrogen or hormones regularly and your potential for getting cancer? Good points and questions. So now we're on to mutation. Want to take a second here and step back and do a little bit of big picture analysis of genetics. I probably mentioned this very briefly at the start of the semester, but it's a good time now that we've seen a few different topics to catch up on where we've been, why it's relevant, and where we're heading. So we started with DNA structure, nucleotides, DNA sequences, we went to chromosome structure, and all of this has led up to DNA replication. If you're going to have chromosomes, and chromosomes are the information that every cell, or almost every cell, not red blood cells, need to produce proteins and to survive, then we need to know how DNA is built into chromosomes, how chromosomes replicate, because chromosomes have to replicate before cell division to make sure that all of the cells that are produced after cell division have the genome, have copies of each chromosome. So where we're at now is finally we've gotten through this is DNA, these are chromosomes, this is how chromosomes replicate to pass on to the cells. The next topic we're going to talk about is what? So we're talking about mutation now, and that is how do chromosomes start to differ? The next topic we're going to talk about is mitosis and meiosis, which is the next logical step. Once you've got chromosomes replicated, DNA synthesis, then cell division happens, and we learn how the chromosomes are partitioned into the daughter cells, how the chromosomes segregate. That's important because at the end of the semester, we wind up getting to talk about transmission genetics, which is the classical field of genetics, which is studying how traits, how disease susceptibilities, how detached earlobes or colored eyes transfer from parents to offspring to their offspring and so forth, right? A really important part of the study of genetics. So before we get there, we have to understand nucleotides, DNA structure, chromosomes, chromosome replication, and then how the chromosomes go from cell to cell during cell division. And that's why we're doing these topics in this order. So it's not, I hope you're not treating each of these topics separately. There's a definite path we're taking from the basics of DNA to the point we're at now, which is how do chromosomes start to become different, mutation. We talk about how chromosomes segregate, and then we can get into the central dogma. What are genes? How do genes produce proteins? How do those proteins affect how we look, how we act, disease traits, disease susceptibility, and then at the end of the semester, segregation of traits in pedigrees, that is parent to kid, kid to grandkid, and so on. So that's where we've been, that's where we're heading, and that's why once we finish with mutation, we're going to move directly into cell division. But not, hopefully, the type of cell division you've heard about before. It is still mitosis and meiosis, but we're going to go at it from a slightly different angle. So last time we got halfway through mutation or so, and here's one other practice. So I threw this at you once last class. So we talked about microsatellite mutation. How would you rewrite this with microsatellite notation? Take a minute. Write it out. Then we'll talk about why we care about microsatellites. So what's the part of this that repeats? What's the unit of the microsatellite? CAG. How many of them are there? So 7, is it CAG? So right, if it's a triplet, then you could call it CAG, right? It could be, or could it? This is hypothetical question. Is it CAG, is it AGC, or is it GCA that repeats? Right, the question is how can you get the most numbers? So this is CAG. If it was AGC, there's no 7th AGC down there, it's AGG. There's only 6 AGCs, there are 7 CAGs, so that makes it the most efficient way to write that sequence. CAG7, and then flanked on both sides by the nucleotides that aren't part of the repeat. Like that. So, mentioned briefly why we care about microsatellites last class. One of the reasons is that they're useful for DNA fingerprinting because each of us, at specific spots of our chromosomes where there are these sorts of microsatellites, 2 nucleotides, 3 nucleotides, 4 nucleotides, that are repeated over and over and over again next to each other in tandem. This is a tandem array of triplets, tandem meaning right next to each other. But we can then use that in forensics to tell who somebody is based on the number of repeats they have at different microsatellites on different chromosomes. There's another reason to care a little bit about microsatellites, and that is that because they change in size from person to person, they can be associated with different diseases if you have too many copies of one of those repeats. And the CAG repeats are in particular relevant to talk about because those are found in a number of genes. Those CAG is also a codon. We haven't talked about translation yet. I know all of you, or most of you at least, have heard about codons and amino acids, and CAG itself encodes the amino acid glutamine, Q, in the single letter code. So there are lots of proteins that are encoded by our genes that have CAG, 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 CAG. Those are called poly for many glutamine repeats. So this microsatellite, this grouping of 3, because 3 nucleotides is a codon, when they're found inside genes, if there are lots of CAGs in a row, they can encode proteins that include long stretches of the amino acid glutamine. 7, 10, 20, 30, 40 glutamines in a row in the middle of a protein. Our genes are meant to have these glutamines normally. The problem is, and we talked about this a little bit during DNA replication, that DNA polymerase sometimes gets lost, so to speak. It loses count of how many exactly CAGs it's replicated when it's copying the chromosome. Sometimes it'll accidentally take one out, sometimes it'll accidentally put one or more in. And that's why different members of the audience class have different numbers of repeats at these different microsatellites throughout our genome. It's a common process that happens. Dinucleotide or trinucleotide repeats, the number grows and shrinks depending on which generation you are and who you are. But there are a series of diseases, including Huntington's disease, so a lot of neurological diseases where people that have these polyglutamine repeats get a few too many CAGs in their microsatellite. So it's something like 20. It depends on which specific gene you're looking at. But there are a number of diseases, a lot of them are called spinocerebellar ataxias. It means it affects your nervous system, it affects your ability to walk properly. Ataxia, can't walk. So individuals that get too many of these CAGs inside genes have proteins then that don't function properly. The proteins can function and do naturally, normally function with a certain number of glutamines in a row. But when you get too many glutamines, then the proteins in the cell start to tangle together, they don't fold properly, and that causes ultimately neurodegeneration. It causes cells in the nervous system to die when they have too many of these proteins that have too many of this one amino acid in them. So a lot of geneticists care a lot about studying how these microsatellites grow and shrink, how they expand in size, increase in size, and then understanding why there's this seemingly almost magical threshold where if you get too many repeats of a dinucleotide or a trinucleotide, 2 or 3 nucleotides in a row, that can lead to diseases. That's one reason to care about microsatellites and to understand that microsatellite notation. Any questions about microsatellites? So that's related to Huntington's disease, spinocerebellar ataxias, and other diseases. So that's one type of mutation. That is, a mutation being defined as anything that changes your DNA sequence. Those repeat expansions or contractions, those are essentially mutations. They cause your chromosome to differ from the chromosome that your parents had. Then we're going to talk about, briefly, a couple of environmental factors that can also cause mutations. So we're going to, in total, then, have 4 different things that can cause mutations. Sometimes, rarely, DNA polymerase accidentally puts the wrong nucleotide in your DNA during replication. That's a common cause of point mutations. Single nucleotide changes, where your parents had one version of a chromosome, one nucleotide, DNA polymerase puts the wrong one in, that's a point mutation. This is another way to get small point mutations or pairs of DNA sequence changes in a row when ultraviolet light hits DNA. So you've been fact-checking, you did already fact-check this idea of how fragile are genes, and a lot of you came up with papers that were related to, yes, ultraviolet light can damage DNA. And it does so particularly by attacking or by cross-linking thymines together. So whenever there are 2 thymines next to each other in our DNA, TT, if ultraviolet hits those in just the right way, then that can chemically bond those 2 thymines together, calling that a thymine dimer, tumor, not T-U-M-O-R, tumor. Not a cancerous tumor. 2 nucleotides connected together. And the reason this is important is because DNA polymerase can't read those 2 nucleotides, the 2 T's, when it's scanning the template strand of DNA during DNA replication. DNA polymerase hits that thymine dimer, and it can't tell what nucleotides those are because they're not in a shape that it expects. That is, they're not individual thymines by themselves, which is what DNA polymerase needs to know how to base pair an adenine with them. So what does the cell do? It's got DNA polymerase, it's trying to replicate this chromosome as quickly as possible because the cell's about to divide and the checkpoints are going to trigger and then the cells are going to get killed if the cell can't replicate the DNA fast enough, and DNA polymerase runs into this thymine dimer.
Does it just add the nucleotides?
So again, cells don't have brains, but this is how the cell assesses the situation. There are 2 things you could do at this point. The cell could either die, which is what happens if it doesn't finish replication, or it can just add in any 2 random nucleotides and hope that they're the right ones so that DNA synthesis can complete so that cell division can proceed. That's what happens during the SOS response. That's what's underlined here, base pairing independent manner. Polymerase hits a thymine dimer. Can't tell what they are. Here are 2 nucleotides. Then keep replicating the rest of the chromosome. So this is a way that mutations can happen. Usually in pairs then, you'd expect 2 nucleotides next to each other that were adjacent 2 T's to be any 2 other nucleotides. They might, by chance, be 2 thymines again. You could calculate the probability of that, I think. What's the probability that DNA polymerase happens to add 2 thymines next to each other at random?
1/16.
Something like that. 1 in 4 chance of getting 1 thymine, 1 in 4 chance of getting the second one. 1/4 times 1/4 is 1/16. So in 1 in 16 times, this works. The rest of the time, it fulfills the cell's purpose, which is to finish replicating the DNA and divide, but it introduces mutations. It enables the cell to survive, but this is a highly error-prone process. But what else is the cell supposed to do? That's all it's got. And then, I hate to break it to you, those of you that are meat lovers, that char on the hamburgers is not always so good for you, much like cigarette smoke. It's been known for a long time that char of most types, especially of organic molecules, that delicious charring on hamburgers, I don't think this works on, what are those newfangled burgers? Beyond Burgers, the all vegan hamburgers. I don't know if, this probably doesn't happen in those. I think you have to be, well, no. Somebody check and find out. Is char on vegan burgers carcinogenic? But diesel smoke, exhaust, cigarette smoke, charring on burgers all have these organic molecules, benzopyrenes. They're carcinogenic. They're mutagenic. They cause DNA mutations. Now, probably not enough that we need to worry about it necessarily unless you're smoking a pack a day or something like that. But this is one of the questions, was how mutagenic are these benzopyrene molecules? So here's how they work. You don't need to know the chemistry behind this. Just trying to show you, these benzopyrene molecules are just polycyclic aromatic hydrocarbons. When they get into our livers have enzymes to try to break down these sorts of toxins. So the enzymes in our livers start to process the benzopyrene, and it's trying to make this, organic chemistry students, if you want to excrete this, then you want it to be soluble in something that we use, like water. So what do you have to do to this to make it more soluble? Add some OH, add some polar groups, hydroxyls. So that's what these enzymes try to do. CYP, in particular, these CYP, CYP enzymes, try to start adding hydroxyl groups to make this a little bit less polar, a little bit more non-polar, so it can be soluble in water and get excreted in urine. But part of this process is it creates epoxides, which are highly reactive. They attach to lots of different organic molecules, and that is how these molecules, as they're being broken down by our bodies, become dangerous to us. And they can react with DNA to form what are called adducts. This is a model of a benzopyrene adduct. It's chemically reacted, that epoxide is chemically reacted with a nucleotide, and it's stuck now in the middle of the DNA double helix, and you can tell that it's making the double helix bulge. So it's not base pairing appropriately. And guess what happens when DNA polymerase comes along and tries to figure out what the hell nucleotide that is? It's got that huge whopping benzopyrene molecule stuck onto it.
Does it just fall off because it's too big?
No idea well, there are a number of repair mechanisms, but the point is it's very similar to the thymine dimer situation in that it can't read what nucleotide that's supposed to be, because it's got this huge polar molecule physically covalently attached to it. So again, polymerase is in the situation where it doesn't know what nucleotide is supposed to go there, and if this is being used as a template strand during replication, then what nucleotide is polymerase going to insert to base pair with it? Probably a random nucleotide. So up to this point, it's been all doom and gloom. Breast cancer, spinocerebellar ataxia, Huntington's disease, but are all mutations bad? Can you take a minute, if you will, see if you can think of a situation in which a mutation, it doesn't have to be a real situation, it could be hypothetical, but I'd love to see 7 or 8 people at least raise their hands after 2 minutes and give me an example of a mutation that would be useful, or at least one that isn't bad, neutral or better. Okay? Take 2 minutes, discuss with a neighbor if you want, Google if you want. We'll come back together and discuss briefly. Okay, what do you think? DNA sequence changes that don't do anything to you or that are good for you? HIV resistance, that would be a great mutation to have, HIV resistance. Related to our current ongoing Canvas discussion, which responses are due by midnight tonight. Okay. Yeah.
So I read an article, I don't remember what disease it has, but in Africa, certain tribal groups, they have a certain disease that's a disease, but that also allows them to not be affected by malaria.
Right. So malaria resistance, does anybody know which gene we're talking about? Hemoglobin. So it's sickle cell anemia. There's a mutation, this is a classic, we might talk about it in class this semester, that if you have 2 copies of the mutation, if both copies of your chromosome are mutated, then you get sickle cell anemia, which is bad. It means your blood can't bind oxygen and transport it to your tissues. But if you're heterozygous, if one of your chromosomes is normal and one of your chromosomes has this mutation, then you're resistant to getting malaria.
What I call the sickle cell trait.
Sickle cell trait, yeah, sickle cell anemia. So that's a fantastic example, right? It's a mutation that is, depending on the circumstance, either bad or good, depending on how many versions of the mutation you have. Yeah? Lactose tolerance, right? Normally, ancestrally, thousands of years ago, humans couldn't process dairy in their guts beyond infancy. It's normal to be lactose intolerant, but some populations of humans got different mutations. Different groups of humans got different mutations in genes that are involved in processing lactose, milk sugar, so that some groups of humans can still process it as adults and not be all farty and upset stomach and gross feeling when they eat cheese or dairy.
The freckles on your skin, I didn't know if it was a mutation or not, but I Googled it, and the first thing that came up was it is partially just UV light, and then also a mutation in the skin cells, and epithelial tissue. So that's why I have freckles.
So are freckles, but are freckles good or bad? Well,
It's moles that are bad.
well, you know, then there are a lot of other different types of mutations we could talk about, because human skin color depends on a series of genes producing pigments in our skin, and if you have mutations in those genes, then you might make less pigment. Scandinavians, for example, or people from the British Isles. Right? It's another example of it depends on where you live in the earth, whether or not it's beneficial, neutral, or bad. Because if you live in places where it's really sunny, it's bad not to have a lot of pigment in your skin, because then you get sunburns and skin cancer, and it's not good.
There are certain codons that will program the same amino acid even though they're different letter sequences. So if it mutates one single nucleotide or maybe two, and doesn't change the amino acid, then it's not a bad mutation.
Right. So when we get to central dogma, we're going to talk about translation and the fact that there are some mutations you can make in a gene that don't do a doggone thing. They're neutral. A mutation changes the single nucleotide that doesn't change the amino acid that's encoded by the codon. It doesn't matter. So that's a great example of a neutral type of mutation. It's not good, it's not bad, it's a DNA sequence change that does nothing. And there are lots.
Do they still get carried down through generations as a mutation?
Yes. So if I have a mutation in my gene that causes the same amino acid to be introduced using a different codon, then that gets passed from me to my kids. It's probably neutral for them too. So we've seen how environment, just a couple quick examples of how environment can impact DNA. So last thing, and then I think we'll end after this. End of last class, I asked you to make a prediction on this type of graph. Now that we know a little bit about mutations and how they occur, we have a gene that's 1,000 base pairs long, a figmental gene, just a figment of our imagination, hypothetical gene, 1,000 base pairs long, at each position on the chromosome on this gene. What's the probability between absolutely sure you're going to get a mutation and zero, no mutation chance at all? What does the probability of getting a mutation in a gene look like? What I'm going to ask you to do is to send me your drawings right now using the Google Classroom assignment that opened at the start of the class. So, again, we're doing this for two reasons. One is to practice annotating PDFs and sending them to me on Google Classroom. The other one is I'd like to be able to anonymously show some of your work up here so we can look at what you're thinking. Instead of me walking around to everybody's device and looking at what you've drawn, I can project some anonymous examples. So if you'll please go into Google Classroom, get that PDF, draw your prediction on that PDF, and send it back to me right now, that would be great. If you don't have the capability of doing that right now, that's fine. This is not for points. This, again, is for feedback. Do it before next class, please. And if I can't see what you've drawn on this, I'm going to send you another private message on Google Classroom that says, I can't see what you've drawn. Please get help from me or from the Discovery Hub.
What is the question again?
So, make a prediction. What's the probability, each spot in a gene, what's the probability that a mutation will occur? Absolutely. So it's going to be probably a line graph. Can you make any sort of prediction about, depending on where you are in a gene, the probability that any mutation is going to occur? It's a weird question, I know. So I'm just quickly going to grab a few examples to look at as you're working on that, and then we'll look at a couple of these examples and we'll finish up. And if anybody has any questions, please feel free to ask. All right. So let's see what we've got. How about this one? So what would this indicate? That near the start of the gene, on the left side, for example, what does this mean? You'd predict that there would be many or few mutations on the left end of the gene. Few. And then by the time you got to the end of the gene, you almost certain probability of seeing a mutation there. Okay, that's a prediction. It makes a prediction about where in the gene you might see mutations occur. Yeah?
So the x-axis represents the gene sequence,
Yes.
and then the 1% probability of it increasing.
So what I intended, and by looking at some of the submissions, it was clear to me that I wasn't clear on exactly what the y-axis is. So this may mean the probability of, by this point in the gene, you've seen a mutation. If you were reading from left to right, so this is not supposed to be cumulative probability. It's just at each nucleotide, what's the probability that it gets mutated? So what do you think? What's the probability? Are mutations common or rare, do you think?
Rare.
They're pretty rare. So the probability of getting a mutation at any spot on a gene, you might predict to be the same at every nucleotide. It doesn't matter which part of the gene you're in. There's going to be probably a low but constant probability of mutation. That is, at each nucleotide in the gene, there's a slight chance that there would be a mutation there. But it probably doesn't matter where in the DNA, where on the chromosome you are, in terms of the gene sequence. Each nucleotide probably has about the same probability of being mutated. So we're going to see next time, we'll get to the case study for the mutation chapter. So for now, we're going to talk about that. We'll finish up. And then, remember, Canvas discussion is due tonight. And for next class, because we're going to get into mitosis and meiosis next class, please try these first things. Short video, the OpenStax readings, and just the first video that's listed in the ones that I suggest watching. Ploidy and the cell cycle.